fish. All right, organizers. Now we get into the big one. This is key to understanding how cells start forming the three germ layers. Because as we've mentioned up until this point, this is where the cells take on different fates. This is where they start going down different pathways, different differentiation pathways. And it's all about the paracrine factors, the juxtacrine factors, the morphogen gradients, and all of those things that they're exposed to during these initial stages of gastrulation. So in reality, organization, or the organizers, or the tissues that are patterning these cells, they're merely groups of cells that are typically releasing paracrine signaling to adjacent cells that tell them what to become. They're typically under autonomous specification, although in some cases there is reciprocal induction going on between the organizer tissue. But we'll find that in each one of these cases, some of the same paracrine factors and methods of organization are found in every single model organism. They're the same, which is why it's so good to study them, because we can learn, again, about how we are patterned. Now remember the axes that we're dealing with. Anterior-posterior is head to tail. Dorsal is the back, ventral is the belly, and then left-right. These are the three axes that we typically look at when dealing with the patterning. So let's talk about Xenopus first. This is one of the more highly studied organizers amongst the four vertebrate model organisms, although chickens have definitely been studied quite a bit too as far as their organizers. So here is the universal concept about organizers. Organizers have the ability to self-differentiate typically into dorsal mesoderm. So in many cases, they can receive their own inductive signal. They're kind of self-differentiating to be able to release, what do we call it, autocrine signaling, as we've mentioned. They have the ability to dorsalize surrounding mesoderm as well as into paraxial mesoderm. So they release various paracrine signals to cause mesoderm to become dorsal. You'll find that these organizers are typically in the dorsal region. They will also are necessary for the dorsalization of the ectoderm, and most key is their ability to initiate gastrulation. In fact, everything we've talked about up to this point, the involution, the ingression of cells, those are all initiated by the organizer in each one of these organisms. Okay? So let's look at how these things work. Now, they've shown that at a certain point, if they remove cells from one part of the tissue, do a transplantation experiment, if they take it from various regions and put it into another region, nothing happens. The cells are not committed at that point to become ectoderm or to become neural tissue or whatnot. However, they find that at a later stage, if they take the same tissue that's been affected by the organizer and move it to another area, it starts forming neural tissue where it shouldn't. So there comes a point where once the tissues or the cells have been influenced by the organizer, they start to become more committed down a particular cell fate, whether they're going to become mesoderm or endoderm or neural tissue, which is what they show here. The same tissue, before it's influenced by the organizer, hasn't committed itself to what it's going to become. So you move it to another area, it's going to become a different tissue. However, in this point, these cells have been exposed and undergone paracrine signaling. You move it to another part, and now it becomes a second neural plate and forms another neural tube. In fact, an extreme case of this, if they take the actual organizer area itself and transplant it to another area, guess what happens? You form two whole embryos. That's how powerful the organizer is it has the ability to pattern the three germ layers even if you move it to another area and you end up getting conjoined Xenopus, or conjoined frogs, essentially, in this case. You get different neurulation events, and it essentially patterns the other part of the embryo as a result. So that's how powerful the paracrine signaling from the organizer is. If you were to take this tissue and transplant it to another area, it would organize a whole nother embryo as a result. So let's look at the fate map of a Xenopus and look at the organizer's influence over that. One of the important things that we've talked about here with the Xenopus is the blastocele. And the reason why this needs to be here is because the organizer, which is typically found right here at the dorsal blastopore lip, as it secretes paracrine signals, if this is confluent or touching the yolk, if there's no blastocele, no ectoderm forms. It actually forms into mesoderm. So whereas these cells would normally take on an ectoderm fate, if you were to remove the blastocele and just put that tissue right adjacent to it, it would form mesoderm. You would get no ectoderm. That's why the blastocele is necessary to separate it so that it's not influenced by some of these paracrine signals that come from the organizer. So let's look at how the organizer forms. There's actually two different areas that we have to talk about in relation to that. There are maternal factors that come into play here that are necessary in the formation of the organizer. In the animal pole, in the vegetal pole of the Xenopus, there is typically a sequestering of a protein called VEG1. And this protein, in combination with another protein, forms a group of cells which are called the Nieuwkoop center. Now, this is not the organizer. However, they showed that the Nieuwkoop center induces the organizer. So these cells release paracrine signals that will then tell these cells to take on that fate of then releasing other paracrine signals. So the organizer doesn't form until the Nieuwkoop center sends out these paracrine signals to cause it to form. And they've shown this through a number of various transplantation experiments on how you get this induction process, which I'm not going to go through for this. However, one of the things I want to show you as far as axis formation is you get morphogen gradients. Here in the ventral region of the Xenopus, you get high levels of bone morphogenic protein, or BMPs, and then you get low levels towards where the dorsal end is at. Here in the posterior end of the Xenopus, you have Wnt signaling. You have high levels of this morphogen factor, and then it gets lower and lower as it gets dispersed through the vegetal pole. So here, the maternal components, you have BMPs on the ventral side, and you have Wnt on the posterior side, and that's how the anterior-posterior axis typically is determined, and the ventral-dorsal axis is typically determined. Now, in order to establish those gradients, we have to first talk about fertilization. I told you this was going to come back into play. One of the things about Xenopus is, as it's set up, in the most vegetal region of the vegetal hemisphere, disheveled gets sequestered. You have disheveled protein. Now, you actually have the protein. You don't necessarily have the mRNA. You have disheveled protein here that's bound. When the sperm fertilizes the animal pole, then you get the cortical rotation. So what ends up happening is that cortical rotation pushes the disheveled to what will eventually become the dorsal region of the embryo. Now, as that protein then gets released, it forms a morphogen gradient where you have high levels of disheveled in half of the embryo and no disheveled in the other half of the embryo. Well, guess what that does? If you remember the Wnt signaling pathway, disheveled blocks a protein called JSK in this pathway. JSK3 is the protein that degrades beta-catenin. So in the area where disheveled is found, you get beta-catenin surviving. You don't get degradation of beta-catenin. Over here, on the other side, where there is no disheveled, beta-catenin is degraded. And that then changes. This kind of creates two hemispheres, and where half of the embryo has beta-catenin stabilized, the other half of the embryo has no beta-catenin in it. Well, guess what? You've already established that in the vegetal pole you have other morphogen factors, such as, let me show it right here, VEG-T and VEG-1. When those cross over with beta-catenin, this becomes the Nieuwkoop center. So here, where beta-catenin and VEG-T and VEG-1 cross, that's the Nieuwkoop center. So guess what happens there? Certain genes are turned on, and then paracrine factors are released, and that induces the organizer. That's how the initial body plan gets set up in the Xenopus is the cortical rotation causes beta-catenin to be moved over to this side, the dorsal side, and then you get this kind of crossover between beta-catenin and Veg-T and Veg-1. That then turns on key genes. If you look back here, turns on SMAD signal transduction pathway, which then turns on other proteins like glucosid. I mean, I don't want you to go through all of the various genes that it turns on. There's quite a few, and it's very complex. But the fact of the matter is, when you get a combination of beta-catenin, which is necessary for the transcription of key genes, and the combination of Veg-T and Veg-1, then you turn on key genes in the Nieuwkoop center, which then initiate transcription of key genes in the organizer. And that's how the organizer gets formed. Another set of genes that get turned on in this combination is called brachyury, or XBRA, and these will induce this region right here to become mesoderm, most of the region to become mesoderm. And so there's a lot of patterning going on here. There's Dickoff, as I told you. We'll talk about Dickoff later on, but that's how it's spelled. It's a German word. Okay, so that's how fertilization actually starts influencing the initial stages of the Xenopus setting up its body plan. So let's talk about what the organizer does. The organizer releases a number of proteins. You can see how many different proteins are happening here. Well, in where the organizer is at, you have blocking of BMPs. So over here, you get this grading of BMPs, but where the organizer is at, it actually blocks bone morphogenic proteins. That's one of the things that it does, is it prevents BMPs from being expressed in these other regions right here. So here, the organizer is going to block the BMPs. BMPs are going to be found all throughout the ventral region here. In fact, the default of the ectoderm is to actually become neural tissue, but when you get BMP signaling, it actually causes the ectoderm to become epithelial, or not epithelial, it becomes the skin and surrounding tissue, so the epidermal layer of the skin and that outer region. So in the area right next to the organizer right here, this ectoderm is actually going to become neural tissue because BMPs are blocked by the organizer. This is a key component of what the organizer is doing. Another thing that it does is it causes the head regions to start being able to be formed. So I'm not going to get into all the details of all of the proteins that the organizer forms. But I want you to know this is also where the dorsal blastopore lip forms. So gastrulation, or the initial movements of the cells, are initiated by this organizer region. It causes the bottle cells to come in, it starts the involution of the cells, and it initiates the epibole that will start surrounding and engulfing the mesoderm and the endoderm from the embryo. So here is just a look at some of the signaling that's going on here. Here, the organizer, one of the proteins that it expresses is Dickoff. If Dickoff is blocked, then the head won't form. But if it is expressed, then you get the head region forming, and you get the neural tissue forming, and so on and so forth. Let's look a little bit about just the two major players in differentiation here. This is where we get into cell specification. The ectoderm, as you should know, becomes not only the outer covering, but also becomes neural tissue. Well, depending upon what the ectoderm is exposed to, there are several fates. For example, if the ectoderm receives both BMP and Wnt signaling, it'll become the epidermis. It'll become the skin. It'll become the outer layer. It'll become, feathers for chickens and other things like that. Here, if BMP is blocked, as it typically is done by the organizer, but Wnt signaling is present, that becomes the spinal column. And in the region closest to the organizer, where you have both BMP and Wnt signaling blocked, that forms into the head ectoderm. And there are various key genes that the organizer secretes, like Cerberus and Dickoff, that block BMP and Wnt signaling. So this is just one example of many different ways in which paracrine signaling will block certain signal transduction pathways. Over here, these are allowed. Near the organizer, they're not allowed. BMP and Wnt signaling is not allowed, and that allows this ectoderm to become neural tissue specifically for the head. Now, one of the things they found in vertebrates that also plays a key role in anterior-posterior patterning is retinoic acid. This is going to come up quite a bit. Retinoic acid is a key factor in turning on many of the Hox genes. And so the Hox genes definitely play a key role in anterior to posterior patterning. So just keep that in mind, that retinoic acid, the gradient, it actually has a low to a high gradient, and that forms the anterior to posterior axis. Let's talk about fish. In fact, this is actually a little bit easier. In the initial stages of the fish or the cleavage of the zebrafish, there is a difference in where the maternal components are. In fact, in this area where gastrulation begins, there's a key gene. Let me jump ahead to it right here. It's called nodal. So nodal is sequestered to this region. If you're looking at the cleavage of the zebrafish here, you can see in this region you've got some sequestering of nodal, where you have a high amount of this protein. This is where the organizer is going to form in the zebrafish. This is also where gastrulation begins, in that embryonic shield, where you start getting involution of these cells. Remember, the organizer is responsible for initiating gastrulation. So here in the dorsal region, over here is the ventral region. This is where the organizer is going to form. One of the key proteins that are responsible for that is NDR1. Okay, let's come back here. So here, right here, this embryonic shield, this is the dorsal region. This is where the organizer forms due to nodal, and this is where involution of the cells begin. We already went through the gastrulation movements here. So let's look at some of the other factors going on here. The same thing occurs. You get combination of beta-catenin and nodal in this dorsal region, and that's going to also create the organizer in that region of the zebrafish. So the same proteins, beta-catenin, nodal is similar to the VEG-T and VEG-1, essentially. So let's look at how this works. That's the anterior-posterior pattern. Here we go, dorsal-ventral. So looking at the top, here's the ventral, here's the dorsal. You can see that over here in the dorsal region is where the nodal is at. This is where involution of these cells is going to begin. You get a pibili of these tissues going around, but this is what's going to start gastrulation and involution. Also, as the hypoblast is forming, they're going to be exposed to various paracrine factors by the organizer, and that's what's going to cause the hypoblast to start forming the mesoderm components and eventually the endoderm components. So the signals received, the paracrine signaling received by these cells as they move through and start involuting is what's going to help them take on their particular fate. Cells or genes are going to be turned on as a result of these paracrine signals. In fact, the paracrine signals change over time as cells that come in late will be exposed to different signals, and they'll actually take on a different fate. Again, retinoic acid plays a key role in anterior-posterior patterning of the zebrafish. You don't necessarily have to know all the dynamics behind this, but retinoic acid, again, is another factor in anterior-posterior patterning. It increases as it goes from anterior to posterior and turns on key genes for that patterning. Left-right. We didn't talk about left-right patterning in Xenopus, but one of the things that happens in left-right patterning in zebrafish is there are these ciliated cells. And due to the ciliation, the cells will actually sweep in a counterclockwise fashion, and it pushes certain molecules over to one side, and that's where you start get the initial formation of left-right patterning. And so this creates a distribution of different genes on one side or another, and that's one of the things they found that will start the left-right patterning that's necessary, is that these ciliated cells will actually rotate in a counterclockwise fashion and push molecules to one side that will then begin that patterning. Pretty fascinating. So these are the three axes. Anterior-posterior patterning is primarily by FGF Wnt signaling with retinoic acid, similar to what you saw in the Xenopus. High amount of Wnt signaling is in the posterior end. Low amount of Wnt signaling is in the anterior end. Retinoic acid plays a key role. As it increases, you get more posterior genes being turned on, and that causes the tail end to start forming, whereas little retinoic acid is in the anterior region, and that forms the head. All right, let's talk about chicken embryos. This one's actually easier than all the others. So, the organizer in the chickens is called Henson's node, after the scientist who actually discovered it. His last name is Henson. So, Henson's node. This is the equivalent of the dorsal embryonic shield for the zebrafish, as well as the dorsal blastopore lip in the frogs. You can see how they all play the same role. They're all for the initial gastrulation movements and the necessary paracrine signaling to start setting up the body plan. So when does Henson's node form? Well, let's look at the chicken embryo. Here is the comparison between the frog, which we just went over, and the chicken. In the orange dots is beta-catenin. So we know how cortical rotation causes the embryo to shift, and then you get beta-catenin in one half of the frog embryo and no beta-catenin on the other. Here's the vegetal pole of the frog embryo, where you get veg-T and veg-1, and then when you get the crossover, that's where you get the Nieuwkoop center. Well, let's look at the chicken embryo. Instead of having beta-catenin to one side or the other, beta-catenin is just around the periphery of these cells, of these epiblast cells that are forming. Here, in the most posterior end, you get crossover between beta-catenin and veg-T and veg-1. That forms the Nieuwkoop center. So same thing here, just this is where the posterior end of the embryo is. Remember we talked about how the primitive groove starts forming from the posterior end? It's right there. It's at this junction, and that's why these cells initiate the gastrulation movements, is because of the beta-catenin and the veg-T that initiate the gene transcription that's necessary for the changes to occur in these embryos. Now, how does the veg-T get sequestered over here? In fact, that is primarily due to gravity. As the egg is going through the oviduct of the chicken, as it's actually rotating. And due to that rotation, the gravitation, it pushes the veg-T to the most posterior end of where these cells are undergoing cleavage. And so that's how veg-T actually gets pushed to this area, and that's what creates the most posterior region where the organizer is eventually going to form from this Nieuwkoop center. So gravity plays a role in the posteriorization and the anteriorization, so anterior-posterior axis of the chicken. So in the actual laying of the egg is part of the anterior-posterior patterning of that. If you just pulled the egg straight out of the chicken, it wouldn't work, well, for a number of reasons. So let's look here. This region, the Nieuwkoop center, will induce the various cells that will eventually become Henson's node, which is the organizer of the chicken. Now, Henson's node doesn't actually form, the organizer doesn't form until these cells reach the most anterior region of the embryo. So as the primitive streak's forming, these cells are being exposed to genes from the Nieuwkoop center, and eventually these cells will take on the organizer fate. Okay? So Henson's node doesn't show up until the entire primitive streak has been developed, and then in the most anterior region of here, you get Henson's node. That's where the organizer forms, is in the most anterior region. Here we have various genes. Now here you're looking on the green region right here. This is the hypoblast. Remember I told you that the hypoblast does not play any role in the germ layers. However, it does play a key role in the patterning process. So here we see we have FGF signaling and Cerberus and other signals that are inducing the epiblast as it starts undergoing its gastrulation process.